Thank you. I'd like to thank everybody thanks everybody for showing up today. So, where's our pointer? Oh, is it? Okay, sorry. Okay.
You could advance, but you can't.
Oh, yeah, no, I'm advancing, but am I not doing that? Oh, there we go. So today I'm going to tell you a little bit about a journey that I've been on for over a decade, and the culmination, or recent culmination, of a lot of hard work and effort on a number of people's part who I'll acknowledge at the end. So today I'm going to tell you about our drug discovery pipeline that we've been working on and developing, and a drug that we have identified that we think might be useful for therapeutic purposes for people with dysferlinopathy, also known as LGMD 2B and R2. But I'm going to call it dysferlinopathy for the rest of this talk, because I think it's really important for everybody to have a handle on what the genetics of their mutations are and the etiology of their disease. Okay, so just a quick overview of what happens when you have dysferlinopathy. You have a mutation, a loss of function in dysferlin, and this loss of function results in an inability to repair plasma membrane damage. And plasma membrane in muscle is called sarcolemma, so I can use that interchangeably, either plasma membrane or sarcolemmal damage. And so in this cartoon diagram that Liz McNally did in 2009, it's still holding pretty well up over time. You see dysferlin, and what dysferlin does is to repair membrane damage. So eccentric muscle contraction, things that put tensile forces on your muscles will rip the membrane. And when the membrane rips, it's a potential for death of their muscle cell. So when the membrane rips, calcium flows in. There's a low amount of calcium on the inside of the cell, higher on the outside. And that calcium coming in is a location marker for where the tear is. And what dysferlin does is it grabs these membranous vesicles that are just below the skeletal muscle membrane and fuses them, sealing the breach. And this is what happens very quickly in people without dysferlinopathy. So unfortunately, if you have loss of function mutations in dysferlin, you get LGMD 2B and dystrophic muscle phenotypes such as inflammation. You have incomplete muscle regeneration. You have massive fat replacement, muscle weakness, and eventually loss of ambulation for many patients. Okay, so we talked about a lot of different therapies in this past few days. And we're sort of on the work that we're doing is sort of on the cusp of a few of these. Very important to our work is personalized medicine. So, how many people out there know they have a missense mutation in their gene? Just raise your hand if you know you have a missense mutation. And hopefully you all know what missense mutations are. If you don't, I'll tell you shortly. And the second class of sort of emerging therapies that we're interested in is this idea of chemical chaperones. So these are small chemicals that can be introduced in the body, and what the hope is they'll restore your mutant protein that's made in your cells if you have missense mutations back to where they belong, and hopefully to functionality. So we're trying to fool the body into allowing these proteins that normally it doesn't want to persist, and if they can, to function. Okay, so let's talk about chemical chaperones and personalized medicine. So when proteins are made in your body, they're translated from RNA into a chain of amino acids that are folded. These proteins have to assume a three-dimensional structure often, or always, to be enzymatically or to be active or functionally active. So they fold properly and then they're functional. But if a protein misfolds, and there are a lot of reasons this happens. In fact, as you age, it's, the proteostasis network is what they try to describe protein folding in cells. That starts to break down as you get older. And so you get more protein misfolding. But these misfolded proteins can be bad for the cell, and so what it does is tied to destroy those misfolded proteins and degrade them or to occlude them into aggregates. And sometimes you see these in many aggregation-prone diseases. Okay, so let's talk about missense mutations. This is the personalized medicine aspects of it. So for those of you who know that you have missense mutations, that's great. Everybody should know what their mutation type is. So I'm using here demonstrating a mutation that is very important in my talk, and also thanks to Simone, who very generously created a mouse model with this particular mutation that's really helped us go forth with our research. So there's in humans and normal humans with human beings with normal copies of Dysferlin, at position 1341 there's a lysine residue, and in dysferlinopathy patients there's a mutant copy, and instead of the lysine it's coded for by a proline. So the lysine residue makes a normal protein, it's folded, it goes and it gets trafficked, and it gets to the membrane surface. Okay, so, but in a dysferlinopathy patient or mice that have a homolog of this mutation, the protein is unstable. It can form aggregates. It also gets rapidly degraded and never makes its way to the membrane surface. It's not there to do membrane repair. So our idea is, well, what if we fool the cell into not destroying this protein, get it to the membrane surface where it can be functional? Okay, so here's within that context of proteostasis in the cell, missense mutations can lead to unfolded and unstable proteins. And we think for dysferlinopathy, about 30 to 40% of the patients have missense mutations. And we know that for almost all dysferlinopathy patients, the protein is virtually absent from the cells. Okay, and that includes all the missense mutations. So a lot of them are being destroyed. Okay, so what is the hope for chemical chaperone therapies? So again, I told you they're small molecules. They're thought to bind to the mutant protein, prevent their degradation, make sure they get to the correct organelle or the right surface. So there are a number of diseases that are currently being treated with chaperone protein, but I'm going to focus on one because I want to demonstrate how this idea of refolding a protein and getting it to the proper place and working is this proof of principle. And that's for cystic fibrosis. And this patients with cystic fibrosis, it affects their lungs, and it affects the cystic fibrosis transmembrane receptor. And there's a company called Vertex Pharmaceuticals that's been very successful in identifying and developing chemical chaperones for CFTR, or for CF patients. Okay, so here's a big slide that kind of does a synopsis of what Vertex has done. So CF is an autosomal recessive disease similar to dysferlinopathy and many LGMDs. And in the case of CFTR, it's an ion channel. It regulates salt and fluid in the lungs. And Vertex has several FDA-approved compounds, and they're all chemical chaperones. And to date, there's about 68,000 CF patients worldwide, and Vertex is making $9 to $11 billion in revenue on these chaperone molecules. So let's dip our toe into this water, shall we? So there are there's 6 different compounds I've listed up here. All of them have chaperone-like activity. The first ones that were discovered by Vertex were ivacaftor and deutericaftor. Caftor just stands for cystic fibrosis transmembrane receptor. So all of them seem to have that suffix at the end, and they have unusual names. But ivacaftor and deutericaftor were able to bind to CFTR and increase its ion flow. And then they came up with these other chemical chaperones that are able to bind to CFTR, and not only so they bind to CFTR, but they make sure it gets to the membrane surface. This is very familiar, right? This is kind of what we want to do with Dysferlin. So in addition to those individual drugs, they can now make combination drugs. So there's lumacaftor and ivacaftor that make a combi. And if you notice below each one, I've showed you how many different missense mutations each of these combination drugs can do. So this is sort of the idea that we have in developing a therapy for dysferlinopathy and perhaps other LGMDs as well. So in the drug discovery pipeline, you develop assays, you screen compounds, you find hits, you promote hits to leads, you do lead optimization, you take those compounds and you try to make them the best compounds you can in preclinical trials before you get them into patients. This is an oversimplification. Many people here know much more than I do about this. So this is a fun part of the story. So I'm a geneticist. I'm a model organism geneticist. And when I started working on Dysferlin, I worked on a roundworm, a microscopic nematode that's found in the soil called C. elegans. Now, the Nobel Prize has been won several times for research in this organism. And we use it as a screening tool for drugs. Turns out that Dysferlin is highly conserved evolutionarily, and it's found in C. elegans. But it's found in the sperm of C. elegans. So in humans, it's in skeletal muscles, and in C. elegans, it's in sperm. But it works the same way. It's involved in membrane fusion events. So here you see the sperm of C. elegans, and what happens is there's a calcium-dependent signal that causes a membrane fusion event. And when that membrane fusion occurs, the sperm develop and are fertilization competent. So we have these mutations in C. elegans in the Dysferlin homolog, and at a particular temperature, the TS mutations that are missense mutations in C. elegans. And at a non-permissive temperature, they're sterile. So we did the screen and just looked for compounds that could restore fertility. And we could do this in 96-well format, and we screened 80,000 compounds. So in the absence of Dysferlin, it's no progeny. If you have a hit compound, it restores fertility. And we found a number of compounds that could restore fertility in C. elegans. So this is one arm of our pathway. We have a number of novel chemical entities that are able to restore this homolog of Dysferlin. But I'm here to tell you that we've tested some of these compounds in our human in vitro systems that I'm about to describe, and they're active. Okay, so this is the beginning of our pipeline. Here's our second screening platform. Basically what we did, and much of this is funded by the Jane Foundation, we modeled 113 different Dysferlin patient mutations into cells, okay, that don't express Dysferlin. And we are able to determine whether these mutations get to the membrane surface or not. We know that they're all pathogenic, or we believe that almost all of them are pathogenic when they gave us a list. And in this system, we were able to identify 64 mutations using a method of analysis called flow cytometry. So we could see whether Dysferlin got to the membrane surface. And we found 64 different mutations where it never did. And we were able to turn this into a screening platform. So we're asking for compounds that, in the presence of the compound, can we get Dysferlin to go to the membrane surface? Oops, Anyway, I can tell you that we found 25 different missense mutations. Okay, can we go back one? Sorry, is that possible? Okay, it's not. So we found 25 different patient mutations okay, I missed this point in the last slide, is that in developing these kind of assays, we needed a control. And we chose a chemical chaperone because of our knowledge of what happened in C. elegans already in the roundworm. We chose this chemical chaperone as a control. And it turned out that control worked pretty well. And it worked on 25 different patient mutations. Not only did it get the mutations to the membrane surface, one of the strongest hits we got was to a mouse model that Simone created of a missense mutation in Dysferlin. And using this mouse model, we were able to dose the animals with 4-PBA and examine their ability to repair muscle. And both in explanted muscles that we treat in petri dishes and from mouse that were given 4-PBA, we could see restoration of membrane repair in a number of these mutations. Okay, so what is sodium 4-PBA? What is this drug? Well, lo and behold, it's an FDA-approved compound, and it's used to treat urea cycle disorders. So it's been prescribed to patients for over 20 years. And what it does in urea cycle disorder patients is it acts as a sink for ammonia. So urea cycle disorder patients have mutations in one of 7 genes. There are 5 predominant ones that are involved in the urea cycle that you see here. Any mutation in one of those 5 will lead to urea cycle and a buildup of ammonia. Well, it turns out that 4-PBA is a drug that acts as a sink for ammonia. So those are the genes. I'm just going to skip that. So this is what 4-PBA does sort of chemically. The drug is called Buphenyl. That's what most patients take. The drug is broken down in the body, and ammonia in the form of glutamine. So ammonia gets converted to glutamine. It gets picked up by the breakdown product of the drug, and then it's excreted by the kidneys. So patients who take this drug take up to 20 grams of it a day. So they just eat it. They put it in their juice, they eat it with their cereal, they have it for breakfast, lunch, and dinner. But it's a life-saving drug. Typically, they test children for ammonia levels when they're born, so the determination of urea cycle disorder is made very early, and those children are put on the drugs and into adulthood. So we've developed these assays. We've got a hit for PBA, and it's FDA approved. And now we started doing preclinical trials. And so I'm going to show you a quick video of what the mouse that Simone created that she allowed us to import it into MIT for us to use as a model for our preclinical work. So as I said, this is one of the strongest alleles that 4-PBA worked on in our assays. And see on the left, you'll see the mouse crossing an inclined beam, and we measure crossing time. So this is the wild-type mouse, and this is a MEX38 mouse with an L1460P mutation, which is the homolog of what's mutated in humans. And as you see, as the mouse gets closer to the end, it has trouble, and it has footfalls, and it takes longer for the mouse to get in. So these are about 1-year-old mice with LGMD 2B. Okay. So in wild-type mice, when you stain them for dysferlin, and you take a cross-section of the muscle, you stain for dysferlin, you see this classical sarcolemmal staining pattern. Okay, so we can see that happens in wild-type animals. But in dysferlinopathy mice, what you see on the top of the screen, you see background staining. Okay, this background staining is just it's called the secondary antibody control. You can ignore it for now. But what you can see is that there's no dysferlin staining with these MEX38 mice that are treated with just water. But MEX38 mice that are given 4-PBA, you can see dysferlin restored to the membrane surface. Okay. When we look at membrane repair, what we do is we explant muscle and we shoot them with a laser beam to create a wound on the muscle, and we track a dye that's otherwise membrane impermeant. So if you don't have damage, it doesn't go in. When you create damage, it does. So this is what happens in MEX38 mice that are just given water. We laser wound, and you can see that the dye goes in and it doesn't get occluded. These are MEX38 mice that have been drinking 4-PBA for a year. You can see the damage. You see that photo bleaching, but it reseals instantly. So we took these mice at the end of the year. We did those kind of experiments with the histology and the membrane repair. But we just looked at just simple muscle weight, and we could see a significant increase in muscle weight in much of the affected muscles in the hind limbs of the mouse. So in the quadriceps, the EDL, the psoas, and the glutes, we see a pretty significant increase in the weight of these muscles. It's not back to wild-type levels, but we see muscle increase. So when we look at balance beam performance, we also see an improvement. So there are different beam widths. I'm sorry, that doesn't look so great on here, but there are different beam widths, and we can track how they perform on each beam width. And what we're able to show is there's a statistically significant improvement in their ability to cross these beams after a year on drugs. And I should say, we just started at a single dose. We really needed to know fast. We didn't do a lot of dose response experiments. This was a whole year assay at a single dose. So part of our next set of experiments is to work on dosing. So So we've done these preclinical studies and we see efficacy in many levels. We think we can get them higher and better, and so we're working on those before we start talking about getting these into patients. Because we'd like to establish some more knowledge about what's happening to the drug in the body of the mice, and if we can get the greatest improvement in the dystrophic phenotypes. And with that, I'd like to acknowledge the number of people who work on this in our lab: Eric Williams, Maddie McGee, Heidi Herbawi, and our amazing dissection person, Won-Tek Chung, who's amazing at doing muscle dissections for us, as well as our funding agencies, especially the Jane Foundation, who have been supporting my work since 2009 on this project. And with that, I will thank you all, the meeting organizers, and there's a QR code if you want to reach out and talk about your particular mutations.